In the next step of in the process of understanding of the reproduction in flowering plants, the fertilization is the process which is followed by pollination. Now, pollination in plants is important, as we have studied, that it is the transfer of the pollen grains from the anther to the stigma. Now, the first step in the fertilization is the pollen pistil interaction. Now, when the pollen grain lands onto the pistil, that is the stigma, there are certain conditions under which only the pollen will be able to germinate, and that reaction enables the pollen grain to develop the pollen tube. So the time from which the pollen grain lands onto the stigma of the flower and develops a pollen tube is known as pollen pistil interaction. However, it is interesting to know that all the pollens that land onto the stigma do not germinate or do not develop the pollen tube. It is because of the compatibility factor. The pollen grains, we know that they are made up of outer layer XII, and this XII is composed of certain chemicals. These chemicals, when the pollen grain will lands onto the stigma, the chemicals which are present on the stigma should be compatible so as to allow the pollen tube to develop. So not all interactions or not all the pollens which are falling on the stigma develop a pollen tube. Also recall that the pollen grain which is being released during the process of transfer is released at two cell stage. That is, one is it is having the vegetative cell one, and the second is the generative cell. Normally, all the angiosperms, they have the pollen grains which are released at two cell stage. So let us now come on to the next step. After suppose the successful interaction, that means the correct pollen grains land onto the stigma of the flower, the pollen grains will start developing the pollen tube. Now here you can see the stigma of a flower where the pollen grains have started developing the pollen tube. Now here, the pollen tube, since the fertilization, that means the male gametes are carried to the female gamete with the help of a tube, we are calling it as a process which is known as siphonogamy. This is the term which has been given when the male gametes are carried from the pollen to the egg cell with the help of a tubular structure. So siphonogamy happens in case of angiosperms. So when the pollen tube starts developing, it will develop along the style. This is the style over here. It will develop over here through the style, and it will move towards the embryo sac. Now, on reaching the embryo sac, we know the structure of the embryo sac, that it consists of the antipodal cells, three antipodal cells over here. These are the antipodal cells. We have two polar nuclei over here. And we have synergids. These are the two synergids. And this is the egg cell. This is the micropylar end, the end by which the pollen tube will enter, and this is the chalazion end. Now the tube has entered from the micropylar end. Now as it is entering, there are certain structures which is known as filiform apparatus. These filiform apparatus are found in the synergids, and these are actually microtubular structures which guide, their main function is to guide the pollen tube towards the egg cell, to allow it to enter. That is, they help in breaking open the pollen tube and allowing the male gametes to fuse with the egg cell. They direct. So here the male gametes are carried within this pollen tube, and over here you can see the filiform apparatus. Now also we have studied that pollen grains are released at two cell stage. So here you can see there are three male gametes over here. The two male gametes and one vegetative cell. How they are obtained? Now during the movement of the vegetative cell and the generative cell over here, the generative cell will divide within the pollen tube to form the male gametes. So here, while reaching the egg cell, this consists of the male gametophyte, that is, there will be two male gametes and one vegetative cell. The function of both we know. Now here, the filiform apparatus, which is finger-like projections over there within the synergid, the pollen tube will enter one of the synergid, causing it to release the filiform apparatus. It breaks open, and this filiform apparatus then guides the pollen tube carrying two male gametes which have been obtained from the generative cell due to the division. We know the vegetative cell role is only to give the nutrition. So here the generative cell forming two male gametes and one vegetative cell, total of three cells, enter inside the egg apparatus. Always remember, when you are drawing the diagram, please mark the chaliceal end and the micropylar end for better understanding and clarity. Now here in the next step, we see that as soon as the pollen tube enters inside or within the embryo sac, here the pollen tube will release the two male gametes, and one of the male gamete was pushed by the filiform apparatus towards the egg cell. The egg cell is pushed towards one corner, and this will try, the filiform apparatus will push one of the male gamete to fuse with the egg cell, whereas the other male gamete is pushed to fuse with the polar nuclei. So here the fusion of the egg cell and the male gamete will be taking place, and this will result into double fertilization. That we will study in the next part of the video. Just trying to recall everything. The pollen pistil interaction is the first step of the fertilization, where it is necessary that there should be a complete compatibility of the pollen grain which is landing onto the stigma of the flower. And if they are not compatible, that means certain chemicals which are present both in pollen and the stigma are not compatible to each other, the pollen tube will not develop. Now, if the correct pollen grain happens to land onto the stigma, the pollen tube will start developing. This is known as siphonogamy, and the pollen tube carrying the two male gametes, that is one vegetative and one generative cell, will start moving from the pollen tube inside the pollen tube, and within the pollen tube only the generative cell will divide again to form two male gametes. So when they enter into when the gametes enter into the pollen tube, they enter with one vegetative and two generative cell. But as they are moving along with the tubule, they divide, and this generative cell will divide to form two male gametes. Now the three cells will move along with the pollen tube and will reach the embryo sac and will reach one of the synergids. As they reach one of the synergids, it bursts open, and the filiform apparatus, which are microtubular structures, push the male gametes and allow the entry of the male gametes within the embryo sac. So they will guide the pollen tube to carrying the male gametes to fuse with the egg cell. Now one of the egg cell will fuse with the one of the male gamete will fuse with the egg cell which is being pushed due to the bursting of the synergid. One of the egg cell, that egg cell is pushed towards periphery and the male gamete will fuse with it. Whereas the other male gamete will fuse with the two polar nuclei which are present in the central cell. This is the structure of the embryo sac, which gives you an idea the location of the various cells which are present in the ovary.